Food tests. Food tests are performed to detect the presence of different components of foods like carbohydrates, fats, protein, starch, etc. In the food test, we add reagent in a food sample. Reagent is any substance or compound which is added to a system to cause a chemical reaction, or it is added in any system to see if the reaction has occurred or not. So there are different reagents which are used to detect the presence of different food components. Sometimes we add food in the powder form. Sometimes food is taken, like whole food is taken, not crushed. And sometimes we use a liquid food in order to detect the presence of different food components. First test we're gonna see is a Biuret test. This Biuret test is used to detect the presence of proteins in any food sample. Biuret reagent is added in this test. So first, as you can see in this picture, in the diagram, here we have taken a test tube, and this test tube contains food sample in liquid form. Biuret reagent, which is blue in color, is added with the help of dropper. Then this mixture is shaken. After some time, we're going to see the result. If the food sample in the test tube remains blue color, it means that there is no protein found in the food. And if it changes its color to purple or lilac or purple color, then it shows the presence of protein in the food sample. So Biuret test is performed very easily, just we have to add Biuret reagent. Next test is a fat emulsion test. This test is performed to detect the presence of fats in any food sample. We have taken a food solution and added few drops of ethanol with the help of a dropper. Then this ethanol dissolves some of the food. Later we add some water. On adding water, if white precipitate is formed, it shows positive result. It means that fats are present. And if white precipitate does not form, or white emulsion doesn't form, then it shows the absence of fats. Third test is to detect the presence of starch. Starch is a polysaccharide made up of many monosaccharides. In this test, we use iodine solution, liquid iodine. It is brown in color. We have taken potato, peeled it from the top, and added few drops of iodine with the help of dropper. Now this iodine, if starch is present in the potato, this iodine will react with the starch and it will change its color from brown to blue-black. If the color of any food sample changes into blue-black, then it means the presence of starch. Starch test can be marred by seeing that whether iodine has changed its color to blue-black or not. Benedict test is used to detect the presence of reducing sugar. In this test, we take Benedict's reagent. So first we're going to take a test tube containing 2 centimeter cube of food sample, food solution, and we're going to add equal amount of Benedict solution. Always remember this. If you are taking 5 centimeter cube of test solution or any food sample, so we're going to take same amount of Benedict reagent as well. After addition, we're going to keep it in the water bath. We're going to heat it in the water bath for like 2 minutes, or we can heat it directly on the tripod on the burner. So if it changes its color to brick red precipitate, then it means that glucose is found in excess quantity. Now, the reason of changing its color is because Benedict solution contained copper 2 sulfate, which is blue in color. So this is copper 2 sulfate is present. Now this copper 2 on reaction with the food sample, which contains glucose, will changes its color to red color. So this copper 2 is reduced to a red precipitate of copper 1 oxide. Now you see there are different colors formed at the end. If the test tube color remains blue, it means no glucose or no reducing sugar is found. If it changes its color to green, then it means traces of reducing sugar. Orange indicates moderate, and brick red precipitate indicates large amount of reducing sugar. Now this Benedict reagent test or Benedict's test is also used to detect the presence of glucose in urine. So like if people who think they are diabetic or who are not sure whether they are or not, or they want to find out if they are diabetic or not, so they can actually they go to the laboratory for urine test. Presence of glucose in the blood can be detected by urine test, by blood test, or by spinal fluid test. So in the urine test, what happens? Glucose presence is detected by means of Benedict reagent test. What happens? Urine is taken in a test tube and Benedict reagent is added, just like same. And then it is heated in a water bath, and if the color of the urine changes into brick red precipitate, it indicates the presence of glucose. And if it shows less if it shows these colors like green and orange, it shows that less glucose is present.